Следующий доклад. Профессор Тревор. Микробная метагеном.
So, thank you, Professor Poltev, and the entire committee for inviting me to meet you all here in Moscow today. And we've already had a quick look around the sites of Moscow. But let's get down to business. I was really surprised about a month ago when I opened up the Economist magazine and saw that the whole issue, well, not the whole issue, but the key of the issue was dedicated to microbes and how the microbiome knowledge has improved so much over these last few years. The topic was Microbes Maketh Man, and some of the artwork I have blown up so you can see how cleverly the front cover was done. I love that image. Man is a superorganism. That is now beyond doubt. So I want to go back and look again at Yehuda and Noel's fragment from 2004, that everything is infectious until proven otherwise. And what I want to do is explore with you the dogma, the new dogma, or the expanded dogma, is that the human microbiome, the infections which make up the human microbiome, cause human chronic disease. And what we can do about it. You know, one of the things that's really strange in medicine is that we really focus on individual diagnoses, whereas so many of the conditions that would appear very different are quite closely linked. For example, here is a study which just came out a couple of weeks ago showing that restless leg syndrome was a very good precursor as an indicator for later coronary heart disease. Another one, that depressive symptoms can be a risk factor or certainly a predictive factor for coronary heart disease. And one just out yesterday, that these high throughput DNA sequencing, which Nikita spoke about, the GWAS studies, can actually detect differences in the subgingival plaque in the mouth microbiome of elderly subjects with and without dementia. Not statistically certain yet, but at least there are links between periodontal disease and dementia. And then, of course, obesity, which is responsible for so many things, is now being fingered for brain damage. Obesity and metabolic syndrome is being linked now with cognitive impairment. Oh, but wait a minute. Obesity can be infectious. The gut microbiome imbalance is catching. Obesity can be communicated. I think some of you may have seen that in the families that you treat. But now the scientific study has shown that to be the case. So we keep coming back to this circle of infection, and that's what I want to explore in the presentation today. The comorbidity wheel is something that we've written about before. This is one of my colleagues, Dr. Prohl, put together this comorbidity wheel from a number of studies that were in PubMed that noted comorbidities along with the primary study diagnosis. And the thing that I myself noticed when I started doing biomedical work back in the 1970s was that the patients we were studying with diabetes often also presented with asthma, rheumatoid arthritis, and hypertension. You think totally different diseases. And yet, as comorbidities, they're very common. This comorbidity wheel, you can actually follow and trace the comorbidities. Schizophrenia, Sjogren's, it's all on the wheel. In other words, maybe these diseases that we think of as being disparate may be coming from the same root cause, but that root cause is so complex that we've been unable to analyse it up to this point. So, way back in the start of this century, we started to work on the basis that, in fact, many of the chronic diseases, the serious chronic diseases, were due to multiple pathogens. Not a single pathogen, but multiple pathogens. And we first found out quite quickly that antibiotics don't work against chronic disease. They may give some temporary relief to the patients for a year or two, but they don't kill the underlying organisms. The only way to kill the underlying organisms seems to be to reactivate the host defences. Because in Homo sapiens, and only in mankind, there is one nuclear receptor, the VDR, which expresses genes for the toll-like receptor 2, as well as the catalycidin and beta defense and antimicrobial peptides. And all of these are essential to intracellular innate immune defences. So if you're a pathogen like Mycobacterium tuberculosis or EBV or CMV that wants to persist inside the cells of the human body, and in particular inside the immune cells of the human body, you have to knock out this receptor, because you can't afford to have catalycidin lying around. So the microbes have to evolve to knock out the VDR. So what we did was come up with a method for reactivating the VDR using a fairly common blood pressure drug, which we retargeted towards acting as the VDR agonist. And these were some early results that were presented in Porto by my colleague, Captain Tom Perez, who had just retired from 25 years at the US FDA. And he went over a retrospective analysis of some of the people that we've been following, patients that we've been following in our collaboration. And the thing that was really amazing was the number of diagnoses that were responding to this one therapy. It's a single drug, monotherapy, ranging from rheumatoid arthritis, Hashimoto's, uveitis, psoriasis, type 2, Sjogren's, down to the where we had very few subjects, polymyalgia rheumatica, myasthenia gravis, diabetes insipidus, with multiple sclerosis there in the middle. Also in Porto, Dr. Greg Blaney, one of our clinical collaborators from Canada, presented 3 case histories. One was a patient with progressive MS, in which the disease was reversed after a number of years, a patient with chronic fatigue syndrome, positive ANA and positive Borrelia, a patient with FM, fibromyalgia and chronic fatigue syndrome. 2 years later in Ljubljana, Greg Blaney also presented some cases on ankylosing spondylitis, autoimmune arthritis, rheumatoid arthritis. And in all of these patients, the endpoint is a simple endpoint. The endpoint is return to work. These are, after all, being treated in the general practice, not in a specialist clinic. And Nikita might, in fact, shrug at the lack of statistics, but the key thing is the return to work. And then last year in Singapore, a colleague from Berlin, Roswitha Goetz-Rapelka, demonstrated a causative correlation between psychiatric disease and immune inflammation using 2 case histories, sarcoidosis and MS. And both of these involved cognitive and memory difficulties which disappeared as the underlying autoimmune inflammatory condition was dealt with. And the endpoint, once again, was return to work. And then just a few months ago in Granada, the International Congress, Norway's Inge Lintef gave 64 subject case series which demonstrated resolution of chronic fatigue syndrome, ME, with this drug. And once again, the endpoint was return to work. So how can it be that one therapy might address so many different diagnoses? Well, it's not all that surprising, because prednisone, for example, has been used in decades in a whole range of inflammatory diagnoses, even sometimes in infectious diagnoses. But But prednisone is an immunosuppressor. It suppresses the immune system. The physicians that are in our collaboration were using a diametrically opposite approach, which was immunostimulation, switching on the VDR again, giving the innate immune system the chance to start working again. The clue to understanding that lies in molecular biology and in understanding this concept, molecular mimicry. We hear a lot about molecular mimicry. I want to try and explain what it is. Just a decade ago, the human genome was decoded. And as Nikita said, there's been a lot of discovery in the last decade, but not much has come of it. But what will come is from one of the biggest discoveries, the one you haven't read about in the media, and that is of the microbes that turn the human body into a superorganism. The NIH Human Microbiome Project ran from 2007 to 2011 and identified millions of genes belonging to thousands of species of microbes which live in and on the human body. For the 25,000 approximately human genes, there are at least 1 million bacterial genes active in the human body, plus the viral genes and the fungal genes. They all interact. When they're within the cells, within the nucleated cells, when the infection is persistent within the nucleated cells, then all the genomes interact. And it's our microbes that make us genetically unique. There is more difference between the microbiome of two individuals than between any other set of markers. There is no statistically relevant or no statistically deterministic rating of illness or health from this, but each person's microbiome is different, and it differs at different parts of the body, and it changes from month to month. In some cases, from week to week, one person's microbiome changes profoundly. So where does this microbiome come from? It comes from our mother. In fact, from our mother and father, but primarily the mother. Babies are born dirty. And when babies are born, they already have a gut full of bacteria. They already have many of the infections that form the base of the accumulation of infections that they'll gather as they go through life. Then, once they start to breathe, they get it from the air. These are air samples. This is from J. Craig Venter research, and this is taken in a hospital indoors in San Diego. And the DNA in the air, there's a little bit of human, about 8%, but most of it is bacteria. In a house, you'd expect the opposite. You expect it mainly to be human. But no, in a house indoors, once again, mostly bacteria, much the same as in a hospital. Not that much difference in infectivity between a house and a hospital, which is amazing when you think of it. But this is what we're breathing. So we're exposed to the microbiota as we go through life. Just about everything we come in contact with is capable of adding to our microbiota. These are the microbes in roughly the proportions that are obtained from smoking a cigarette. Pseudomonas, Cinebacter, Klebsiella, Clostridium, down to Staphylococcus. And then, of course, food. This is a study that was done by Eric Allen's group at MIT. And what he did was he looked at the genes that were connecting the human microbiome genes, with sources of the genes, in this case farm food, soil, and hydrothermal heteropod. But the key thing is the red areas, because it's a heat map, and you can see there's a significant correlation between genes that are part of the human microbiome and the genes which are present in the food sources. So we get it from food. And of course, over the last few decades, the amount of travel and international transport of food has risen immensely. Populations have different microbiomes. The urinary metabolome, this is early work done by Jeremy Nicholson, and the urinary metabolome you can actually measure in the urine of different populations different sets of metabolites. I was going to say peptides, but it's peptides, proteins, and other metabolites as well. And they're unique to the area. So if you shift people from, for example, America to Japan, then you suddenly find their metabolome changes as well. And from reverse, from Japan to America, there's a Japanese American in there. So the metabolome varies as we go through life, and there are so many sources that it varies from. So I said that the important area is within the cells, because when you start looking at the microbes that can actually exist within the cells, then you're looking at the microbes that can interact with our own genome. You can look at the interactions both at the level of the proteome of the pathogen, but also the RNA from the pathogen is also going to impact the way that the human DNA is transcribed. These are TEM photographs, electron microscopy photographs, that were taken at Columbia University back in the 1980s, and they show infection using a uranyl acetate stain. They showed nuclear material, which was due to bacterial infection, in cells from the eye of patients with various diseases. This one is a monocyte from a sarcoidosis patient. And you can see hundreds of tiny coccoids in the colonies which have parasitized this cell. And the important thing to notice is that they're colonies. You're dealing with colonies. The microbiota form into colonies. They don't necessarily, each species have to have all of the genes they need in order to survive. Only the colony needs to have the genes that are necessary in order to beat down the human immune system. And it's the human innate immune system which is active within the cells. Another shot from the Roscoe study. This is a juvenile rheumatoid arthritis lymphocyte. Once again, with a very large infection here, which, if you were looking at it through an optical microscope, you'd probably say that was a vacuole, because you wouldn't see the nuclear material staying so dark. And then a number of other infective regions around the phagocyte. When you look at it with an optical microscope, this is actually a monocyte which has exploded. The infection in the cytoplasm is so great that I'll take it back to the beginning and you'll see it again. The infection in the cytoplasm was so great that the monocyte, the monocyte has actually exploded, throwing out these filopodia, hundreds of little filopodia, very, very long, thin tubules, which are also characteristic of microbial propagation in man. So it was pretty obvious that in order to study molecular mimicry, in order to study how these microbes could possibly cause so many diseases that we see in chronic disease, we needed something else. And we sat down back in about 2003, 2004, and produced something called a computational microscope, which was capable of studying how these molecules interact within the cell at the level of the molecule. 100,000 times smaller than the cell, smaller than the electron microscope is capable of seeing. And this type of microscope is possible because chemistry defines how the molecules are put together, physics defines how they exist with relationship to themselves and with each other, maths sorts it all out, and of course supercomputers do all the computing. The software we used to tie it together was some software called GROMACS. And with that, we can actually simulate molecules. We can go to the genome of E. coli, for example, go to the E. coli genome and pull out from it a gene, in this case a gene called YDGG, and then use the molecular microscope to fold that gene and then see how that gene interacts with the drug. On the left we have a human angiotensin II receptor with a drug olmesartan in the binding pocket. And on the right we have the protein YDGG from E. coli genome with olmesartan in the binding pocket. Now you can look at these gene sequences and say, oh, their homology is very close. But when you fold them into 3D space and see how similar they are, you can understand why the human being is going to have so much trouble when it's got that sort of protein floating around inside its cells, trying to figure out what to do with it when it's expecting to have human angiotensin receptor instead. The other thing that's really interesting is there is a drug bound into this. The drug, the human drug, affects bacterial proteins as well as human proteins. Very interesting. Actually, when you dig closer, this is E. coli again, when you dig closer you find that one of the reasons the proteins are so very much the same is because they do the same job. They have co-evolved or pre-evolved. In this case, you've got the glucose metabolism from E. coli, which starts off with glucose 6-phosphate at the top, finishes with pyruvate at the bottom, just like mankind does, just like Homo sapiens does. Glucose 6-phosphate down to pyruvate, and each of these genes along the way are almost identical to the same genes in man, and they do the same job. So when they become mixed up in the cell, when the genes become mixed up in the cell, when the RNA, when the protein becomes mixed up, this molecular mimicry goes to the very next level that mankind just can't figure it out. Another way of looking at the similarity is to plot a Manhattan plot, and this shows the homology between the Fusobacter genome and the human genome. Now, the homology means the degree of similarity for each of the genes in the Fusobacter genome, and you can see some of them are very, very high indeed. That's normally a line which one uses to say that they're essentially functionally equivalent. But you can see there are a lot of genes in Fusobacter that are very, very similar to man. So if there's a Fusobacter infection, it's not just one thing that it's going to be changing in the way that Homo sapiens works. It's a number of things in Homo sapiens that it's going to be changing. The microbiome messes with our human biology. Inside each cell, many, many human metabolites are affected by genes from the microbial metagenome, and the sum total of all these interactions gives rise to the symptoms suffered during chronic disease. The sum total. So you have genes from bug A plus bug B leading to a dysfunction X. That might be a cardiovascular dysfunction of some sort. Genes from a different set of bugs minus genes from a species G here, which would be competitive with these, because not all of the microbes can cooperate. Many of them are competitive. It might lead to another dysfunction, for example. So genomes accumulate gradually during life, and it's genes from the accumulated metagenome, when you have the wrong mix of genes that happens to give a dysfunction that the body can't manage. You, at that stage, chronic fatigue syndrome, myalgic encephalomyelitis, normal controls, and neurological post-treatment Lyme syndrome patients. Well-defined buckets. The thing that's interesting is that out of about 2,700 total proteins that were examined, 1,600, only 1,600 were common to all three groups. And that 738 were unique to the chronic fatigue syndrome diagnosis, 724 were unique to the normal, healthy, and 692 were unique to the neurological post-treatment Lyme diagnosis. We're not talking about one pathway going wrong. We're not talking about the JAK-STAT pathway going wrong or the nuclear factor kappa B pathway being disturbed. We're talking about lots and lots and lots of things going wrong all at once. And miraculously, the human body normally manages to balance those. When the microbiome overbalances the ability of the human body to regulate, that's when we get dysfunction that we can diagnose as disease. This innate immune weakness is unique to Homo sapiens because it is only in Homo sapiens that one nuclear receptor, the VDR, expresses the genes, the beta-cathelicidin and beta defenses. In the mouse, for example, VDR has nothing to do with cathelicidin or beta defenses because the mouse has evolved differently from man. Man has a very unique innate immune system, and that's one of the reasons why it's so hard to get disease models for immune disease. Let's take some of the pathogens that we've already studied. EBV, Epstein-Barr virus, is being blamed at one time or another for just about every autoimmune disease, I think. EBV is one of the classic ones. That really knocks down the VDR expression, especially in lymphoblastoid cell lines incubated for 2 months or 1.5 years. 15 times it knocks down VDR expression, gene expression. Very profound effect. But other species also commonly associated with these inflammatory diseases, Mycobacterium, Borrelia burgdorferi, Chlamydia trachomatis, Aspergillus fumigatus, HCV and CMV, they all are known to knock down the VDR by a large number, more than 5 times in expression. So when you have a cell which is already infected with EBV, already infected with MTB, and along comes Shigella or some other species that would normally have trouble living in that cell, the microbiota can accrete because the cell's immune system has already been weakened. The innate immune system of Homo sapiens is weakened as we travel through life, as the microbiome builds up and collects more and more species. So what can we do about it? We looked at this VDR and tried to figure out, well, if we put a drug in the binding pocket, then how can we try and make sure that the VDR becomes activated again? And that was a short sequence from our computer microscope, which basically showed the dynamics of the interaction between the two molecules. You can see the ligand and the receptor are both moving. These are two key key groups on the receptor for activation. It's actually possible with a computer microscope to get a very good idea of what's happening at the molecular level. And we were able to find that this drug that we knew had something to do with these disease states because it had been reported by patients that they were getting adverse reactions and in some cases positive reactions from this drug. That this drug was working by reactivating the innate immune system. This drug all of a sudden reactivates the VDR, and by doing that it reactivates innate immunity. Now normally within a body you've got the microbes and you have a steroid, calcitriol, and that calcitriol in typical healthy individuals activates VDR. However, when the microbes secrete substances such as gliotoxin, which comes from Aspergillus, that blocks the VDR. And it stops the VDR from being activated. And you can't use calcitriol to reactivate it because calcitriol hits so many other receptors throughout the nucleus, throughout the cytoplasm as well. So you have to use a drug. In this case, we used olmesartan to activate the VDR, which then reactivates the translation of the VDR genes, and the innate immunity is restored. We retarget it by, instead of dosing the drug once a day, by dosing it frequently and allowing a basal concentration to build up in the bloodstream. That reactivated the drug to become a nuclear receptor agonist. Because the drug was already approved and safe and in use by tens of millions of people around the world, we were able to go straight to patient-important outcomes, put it straight into clinical collaboration. And the results I've already spoken about. So let's summarise at this point. What we found that was that the molecular mimicry as well as the inflammation both need to be treated. In other words, if you use an immunosuppressant to get rid of the inflammation, you'll get rid of the cytokine storm, the autoantibodies to some extent, the nitric oxide, the ROS, barn and urea that come from the nitric oxide, the reactive oxygen species, blood urea nitrogen. But you won't get rid of the molecular mimicry, the interactome damage, which leads to body dysfunction, the cortisol axis dysfunction, the thyroid axis dysfunction. You've got to deal with them both, and the only way you can deal with them both is to actually get rid of the microbes. But attacking the inflammation, or indeed the mimicry, if you could, at any level, is only part of the problem. You've got to get rid of it by dealing with the microbes. But when you do that, you generate immunopathology. Because what happens is your host resistance mechanisms act on the pathogen. Some of those host resistance mechanisms cause damage to the immune system of the host itself, but also there is direct damage by the pathogen. This damage that falls back due to the resistance is immunopathology. It's the immune system damaging the host. When you kill bugs that are inside a cell, when you kill microbes that are inside a cell, you will lose cells. Cells will die as the microbes die and release their DNA and cytoplasms inside the human cell. And when you do that, the patient gets ill. The patient can get very ill indeed, in fact, with immunopathology. And here is a couple of examples from our clinical cohort. This is a rheumatoid arthritis patient, started with an ANA titer of 1 to 160. That doubled to 1 to 320 at about the 200-day mark, at about 9 months. And then it took around 900 days before the ANA titer had dropped back to 0, and it stayed at 0 after that. But the disease gets worse. The disease symptoms gets worse as you start to kill these microbes, as you enable the immune system to kill the microbes again. The disease gets worse. This is an ankylosing spondylitis patient. You can see the sedimentation rate rises to day 600, about 2 years. The C-reactive protein rises, and the Bath Ankylosing Spondylitis Disease Activity Index, BASDAI, in fact, doesn't start to fall off until about the 2-year mark. We're talking about really long-term therapies. This is not something that a person will walk into the clinic and say, gee doc, I want to get cured, and is then going to be told, well, it's going to take about 5 to 10 years, because that is what we're finding it takes. And initially, for the first couple of years, things get worse. So how does this fit into the need for predictive and preventative medicine? Now that we actually have a better understanding of the infections that drive the disease model in Homo sapiens, then the importance of early treatment becomes clear. We can also talk about avoiding microbiome contact, not going into public places, for example, not travelling, not eating. That doesn't work very well. So you can do a certain amount of avoidance, but the main thing you can do is early treatment. Once the immune system starts to succumb, then treatment has to be brought on early. And I've suggested VDR dysfunction, which can be measured. There are metabolites that are easily measured to show when VDR dysfunction starts to occur. Early autoantibodies or a comorbidity. Because remember, look, if restless leg syndrome leads in a reasonable number of cases to coronary heart disease, why not start treating the patient's immune system when they present with restless leg syndrome, before the infection has overwhelmed the immune system? Because a person with no inflammatory dysfunction suffers no immunopathology at all. This drug we're talking about is prescribed for tens of millions of patients around the world as a blood pressure lowering drug. Most people have never experienced immunopathology with it. A small number have had bad adverse reactions and changed to another ARB, but you know, if the person is basically healthy, the body can handle the extra immunopathology. So we're back at the beginning. Everything is infectious until proven otherwise. We have to make a decision. If we think everything is infectious until proven otherwise, why are we treating it with immunosuppression? Because we normally would not. suppress the immune system from attacking the microbes that are causing the infection, that are causing the disease. The palliation of symptoms usually makes the underlying infection worse over time. Now, we can now routinely find microbial DNA in healthy human blood. Trust me, we've done it at Murdoch. Everybody that's looked finds it. The clinical paradigm that we're using really must adapt to that. We really must understand that we are a superorganism, and there is a choice to be made between feeling good with a palliation of symptoms, immediate relief of symptoms, and health, long-term absence of disease. And the two are not necessarily able to be achieved at once. Once you understand that the microbiome are microbes which are going to increase in number and virility under immunosuppressive regimes, it becomes treatment and preventative medicine becomes very much more difficult. Now, since Nikita talked about SNPs, I'm just going to do this one final slide. Keeping you away from lunch, I'm sorry. But we're on time. We'll do this one final slide. Yeah. Which talks about the 10 billion SNPs that have been found in the human genome. And you know, I told you that anybody can find human SNPs in blood, microbial genes in human blood. So when somebody takes a sample from the patient and sends it away for sequencing, they're sending away a mixture of microbial DNA or microbial RNA and human RNA and DNA. And in fact, what you get back is SNPs and mutations which don't really exist. They're due to molecular mimicry, the similarity between the microbial fragments which are going away to the GWAS and the human fragments that are going away. Now, there are tools that will get rid of the microbial DNA component. This is one from Barry Merriman and all. The BFAS is an assembler which is very resistant to errors, very resistant. And then the Edwards Lab blog at SDSU, because Argonne National Labs, Department of Energy, actually has a website that you can send huge gigabytes of data up to, and they will sort out all the microbial data and then send you back to human. So that when you do your own SNP analysis, you end up with the right number of SNPs and not by looking at microbial genes as well as human genes. So there, I hope that was helpful. And now I think it's time for lunch.
Great, nice, fantastic. Thank you, Marco, for not only interesting, but stimulating, fruitful, hypothesis-driving, hypothesis-generating lecture.